Well, welcome everyone to our second lecture on microbial genetics. As Dr. Shane just said, we'll be discussing a bit more about translation this time, and then some gene regulation. So, there we go. So just a brief review of our previous lecture. I hope you all will recall that last time we discussed a bit about DNA replication, so how this DNA strand becomes replicated and then separates into two strands to be placed into its daughter cells. And we also discussed transcription, which was how the DNA is converted into a molecule of RNA. Today, we will be discussing some more about translation. This is how this RNA is then used as a template in the synthesis of a protein, or more accurately, a polypeptide chain. I thought just for those of you who are interested in some more advanced microbiology, I would just mention the process of reverse transcription, because this is a process you will be encountering later on in your career as you keep studying microbiology. So reverse transcription is a process where the reverse transcriptase enzyme is used to generate a complementary DNA, so cDNA from an RNA template. So it's the exact, as the name suggests, it's the exact reverse of transcription. And this process is used by certain viruses such as HIV and hepatitis B to replicate their genomes. But that isn't important for today. Just thought I'd mention it. So just to give you a brief overview of this central dogma of biology, which is the topic of these guest lectures that I'm presenting. Just as a reminder, this is how we're going to move from a DNA. We're going to go through transcription to an mRNA, and today how we're moving from the nRNA via translation to a polypeptide. So this polypeptide, just a formal definition, it's a linear organic polymer consisting of many amino acid residues bonded together in a chain, forming a part or a whole of a protein molecule. That last part is pretty important, because here you can see this is your polypeptide chain comprising these many amino acids. And it's important, it's an important technicality where the end of translation results in a polypeptide chain, not technically always a protein, because it is possible that these chains still need to be connected together in order to form a whole protein molecule. So technically it's more correct to say the end of translation results in a polypeptide chain, not necessarily a protein. Some other definitions you need to be aware of, and I hope you remember a lot of this from a previous lecture. So codons, as you can see over here, these are trinucleotide pairs. And in this case, they also the tRNA also has anticodons. You'll see that these are complementary to one another. And then the tRNA uses these anticodons that will be bound to the codons in the ribosome, which we'll see in a second. And that is how the order of your amino acid chain and the type of protein that is produced will be determined. So if you recall, DNA is our original template strand, which was used in the formation of your mRNA, and the base pairs on your mRNA will influence how these anticodons are bonded to it, which will influence the order of your amino acid chain. So DNA, this is how DNA is actually influencing how your proteins are structured. And then just because I kept hampering on about this difference between a polypeptide and a protein, so just the definitions. A protein is a functional, fully assembled macromolecule in a cell. And it is possible, like I said, some proteins can consist of a single amino acid chain, but others may include more than one. Whereas a polypeptide chain is just another term for a single amino acid chain. It can form a protein by itself, but it might need other polypeptides. Moving on to our codon chart. So recall that the codons are what we find on the mRNA, and it's a trinucleotide sequence that corresponds to a specific amino acid. Just a reminder from our previous lecture, you'll notice that there are U's on this codon chart because we're dealing with RNA. So the thymine, the T, has been replaced by uracil, the U. So there are 61 codons that code for amino acid, and each is read to specify a certain amino acid of the 20 commonly found in proteins. So there are a few exceptions here where AUG is always your starting codon, and it codes for methionine. And there are 3 more codons that don't code for amino acids. This is UAA, UAG, UGA. They are known as stop codons. So they are used during the completion at the end of the synthesis of your polypeptide chain. So just as a brief example to show you how this chart works. So if you are given the codon GAC and you're asked to determine what amino acid it would code for, you would look at the first position. So that's G. It puts you in this bottom column down here. Then you'd move on to A at the second position, putting you over here. And then you'd look at C. There's your GAC. And that codes for aspartic acid. Something that I just like to point out here is please note the number of codons that code for the same amino acid. Look at lysine here, for example. There are 6 codons that code for lysine. This is important when we move on to what wobble is, because that is a consequence of wobble. So the wobble position in a codon refers to the third nucleotide in a codon. So here you can see these UG regions, the third one, the third nucleotide is the wobble pair. And it has 2 major consequences. So the binding of a codon in an mRNA to its cognate, or a member corresponding tRNA, is much looser in the third position of the codon. So this allows for non-Watson-Crick base pairing to occur in this position. Also, it means that there's some redundancy in your genetic code, meaning several different codons can code for the same amino acid. Remember the lysine that I just showed on the codon chart, how there were multiple codons that coded for the same amino acid. So the net effect of all of this is that wobble allows mRNA to be translated with fewer than 64 tRNAs that would be required without wobble. Looking at the structure of tRNA, so the tRNA molecule has a distinctive folded structure with 3 hairpin loops that form the shape of a 3-leaf clover. So here you can see are the 3 loops that they're talking about. And on one of these hairpin loops, you will find the anticodon. So down here, remember this will be paired with the codon on the mRNA. And at the opposite end, so this is where your amino acid will attach. You can see here A is the amino acid is attached here. And then, so after the amino acid is attached, then it'll move on to the ribosome to be paired with the codon on the mRNA. So how are these amino acids activated? So amino acid activation is also known as amino acylation or tRNA charging, and it refers to the attachment of the amino acid to its transfer RNA. This is catalyzed by amino acyltRNA synthases, of which there are 20, and each is specific for a single amino acid and for all tRNAs, which may be properly attached. So these cognate tRNAs. This results in your tRNA taking this shape where you have your anticodon region at the bottom, your tRNA molecule in the middle, and then your amino acid at the top. So just to clarify, this anticodon region is still part of your tRNA. So your tRNA with the amino acid attached to it, and all of these will now be moving to the ribosome so that the protein or the polypeptide chain can be synthesized in the ribosome. So moving on to our ribosome. So the ribosome is the site where protein synthesis will actually take place. A unique distinction of bacterial ribosome is that it is a 70S ribosome that comprises of 30S and 50S subunits. So here you can see this is the combined 70S ribosome. And it, like we said, it comprises of a 30S subunit, which contains 16S rRNA and has more than 20 or 21 proteins or polypeptides, and your 50S unit, which contains your 23S rRNA and it has more than 30 polypeptides, so 34 in this instance. So what is the role of this ribosome during translation? So what is the role of ribosomal RNA in translation? So ribosomal RNA constitutes the structure of the ribosome. And then we're also going to talk about what the 16S RNA function is specifically. So it binds the shined Algarno site on mRNA for protein synthesis initiation. and it also binds protein needed for the initiation and translation of amino-acyl tRNA. So just some more information of the Shine-Dalgarno site, because it's a intimidating name, but it has a very important function in that it's a hexamer that comprises the sequence AGG, and it's a ribosomal binding site in archaeal and bacterial messenger RNA that is located upstream of the start codon. Remember that start codon is always AUG. And the function of this sequence is it helps to recruit the ribosome to the messenger RNA to initiate protein synthesis by aligning the ribosome with the start codon. So it helps to manipulate all the components required for protein synthesis and get the correct ribosomal get the ribosome to the correct starting location to start translation. And then the 23S rRNA has the ribosome that catalyzes the peptide bond formation. So this peptide bonds that they refer to here is the bonds between your amino acids. So these are the bonds on your polypeptide chain. So the 23S rRNA helps catalyze that bond formation, synthesizing your chain. So how is the protein synthesis then initiated? So this involves the ribosomal subunits and numerous additional molecules. One of the important ones is N-formylmethionine, also known as F-met. You can see the F-met over there, and I'll be referring to it as F-met from now on just because it's simpler. And it's a derivative of amino acid methionine in which a formyl group has been added to the amino group. And it is specifically used for the initiation of protein synthesis from bacterial and organellar genes. That last part is pretty important because F-met plays a crucial role in protein synthesis of bacteria and mitochondria, which you have, and chloroplasts, which, of course, plants have. As you continue on again with microbiology, you'll also start learning more about why there are similarities between mitochondria and chloroplasts and prokaryotes. It's pretty interesting when you get into it. So moving on to, or continuing on with protein synthesis. So in bacteria, initiation begins when initiator codon binds the 16S rRNA in the 30S subunit, and the Shine-Dalgarno sequence of the mRNA is aligned. So here I'm just going to show you at the bottom, this is where the Shine-Dalgarno sequence is. Note that it is a region on the mRNA and that is located upstream of the UAG, so the start codon. So this is how your ribosome knows where to start. The 30S subunit is aligned over here, and then transcription will happen in that direction. There are also 3 initiation factors in bacteria, aptly named initiation factors 1, 2, and 3, which play a role during the initiation phase of protein synthesis and help with the assembly of the initiation complex, which we'll be discussing next. And then GTP is what is used as a catalyst here. GTP stands for guanosine triphosphate, and it's a purine nucleotide triphosphate and acts as the energy source for these processes. So as I said, we'll be discussing the formation of the initiation complex. So the key players here, here's your 30S subunit, your mRNA, these IFs that you'll see over here, and the F-met and the IF3, these are all your initiation factors. And then here's your 50S subunit. So going through how this complex is built, first the Shine-Dalgarno sequence, the AGG interacts with the RNA molecule and anchors the 30S ribosomal subunit at the correct location on the mRNA template. Then initiation factor 3 agent binding the mRNA to the 30S ribosome. F-met, remember this is the amino acid used for the initiation. Its binding is mediated by the initiation of initiation factor 2 it's mediated by initiation factor 2, sorry. And then initiation factor 1 binds, causing the release of initiation factor 3, creating the 30S initiation complex, which can now bind with the 50S. So everything that I just described happens over here, and this is your end result. The 30S initiation complex is now bound to the mRNA, and now the 50S unit can be added on top of it. So when this 50S unit is added on top, binds on top of it, initiation factors 1 and 2 are released, and then the 70S initiation complex has now been completed. Something that I'd like to point out here that you just want you to keep in mind are these sites. The A site, the P site, and the E site, these are all sites on your ribosome, which I will be giving you some more detail on here in a slide or two. So once this this initiation complex has been built, the polypeptide chain formation can occur or can start. So this is the elongation of this protein, the amino acid chain over here. So during translation elongation, the mRNA template provides the tRNA with binding specificity. So in this image down here, you can see the mRNA, which is this long molecule down here, provides the temporary binding sites for the anticodon. So the codons down here, that 3-AGU, remember that's our usually our starting sequence, will provide binding sites for the anticodons on the tRNA. So in this image, the sites have unfortunately been flipped. So the A site here would be on the side, and then P and then E. So the A site involves the recruitment of your tRNA with your amino acid. Your P site is where this amino acid forms a polypeptide bond with the existing chain, that's the growing protein or the growing polypeptide, and then your E site is where your tRNA will be expelled once it's deposited its amino acid, essentially. So again, as a reminder, GTP is what provides the energy for these reactions. And then note that once this tRNA moves out, it will leave behind its amino acid, and that is how the amino acids are, or how the structure, how they form. So moving on to these binding sites that I discussed, this diagram I thought illustrated it more clearly. So here you have your A site, your P site, your E site. So your A site is your amino acyl or your acceptor site. This binds the incoming amino acyl tRNA. So here you can see this tRNA is coming in with its amino acid on top of it. It's going to move into this A site, binding to its codon region. So the codon here is UUC, and then the anticodon on the tRNA will be AAG. So this will form a temporary bond before moving to the P site. The P site is the peptidyl or donor site, and this binds the initiator tRNA and forms this polypeptide chain. So here you can see this MET is what came from this original tRNA. This is the methionine, and there's your AUG, or your starting codon. So the starting one has already been kicked out from the E site, the exit site. So this briefly binds the tRNA before it leaves the ribosome. But note how this amino acid has now remained behind, and this is how the chain is forming. It's been bound to the following amino acid. This TRP will then be bound to this PHE, and so on and so forth until this chain starts growing, and that is how elongation occurs. So at the end of elongation, we have to terminate this protein synthesis, and this takes place at one of the 3 stop codons. Remember, I mentioned them earlier. This is the UAA, the UAG, and the UGA. And what happens here is, upon aligning with the A site, so you can see in this diagram over here, just keep in mind the A site would be approximately over there. So this is just during the elongation phase, how this ribosome or the mRNA is moving relative to the ribosome. And here is your stop codon is approaching on the mRNA, and now it's in the A site. So this is what we'll be describing once it hits this A site. So these codons are then recognized by protein release factors that resemble the tRNA. The release factors instruct the peptidyl transferase to add a water molecule to the carboxyl end of the P amino acid. And this reaction forces the P site amino acid to detach from its tRNA, and the newly made protein is released. So that's what happened here. Now, see, now this the polypeptide chain or protein, polypeptide chain is the better term, has been released from your complex, from your ribosome, and then after this has been completed, the ribosome will then also just detach from the mRNA, and it can move off to play a role in further the decryption of other no, sorry, translation. Sorry, translation. I knew I was messing that up. Translation. Remember, you can always distinguish between the two. Transcription happens before translation because C becomes before L in the alphabet. Easy way to remember that. So yeah, the large and the small units dissociate from the mRNA and from each other, and they can then be recruited almost immediately into another translation initiation complex. So just a brief overview of our entire process so far. So here's our start codon, the AUG. The components to begin this translation then come together. And this happens when the initiator tRNA anticodon recognizes start codon. So our anticodon recognizes the start codon, and the mRNA is bound to the small ribosomal subunit. The large ribosomal subunit then joins the small unit, and a second tRNA is recruited. That's what's happened over here. And then as the mRNA moves relative to the ribosome, successive tRNAs move through the ribosome and the polypeptide chain is formed. So this is what happens through these steps, how these the polypeptide bonds form, and this protein just grows and grows and grows. And then this will happen until the stop codon is reached. See here, UGA that's another stop codon, and it's in the A site. And then once it enters the A site, a release factor is triggered and termination of translation occur, and the components dissociate. So here you can see the large ribosomal subunit, the small one, and the polypeptide chain, or in this case, as they refer to it, as a new protein, can all be released. So now we're moving on to some gene regulation, and why is gene regulation needed? So let's quickly use a terrifying eukaryotic example. So imagine if your eye cells produced the same hydrochloric acid as stomach cells. This is theoretically possible as both of the cells contain your DNA, but it would obviously not be ideal. You don't want acid in your eyes. So therefore, genes and gene expression must be regulated. The same holds true for bacteria, where they don't want all their genes expressed all the time. So there are two manners of gene regulation that we're going to be discussing now, and that's inducible genes. These are genes that are off by default and can be turned on by the presence of a particular molecule. And repressible genes are their inverse. These are genes that are on by default but can be turned off by a small molecule. Their regulation will depend on environmental change and the presence of a co-repressor and repressible genes. We'll get into that a bit more here in a slide or so. And then I thought I'd mention that housekeeping genes are genes that are constantly expressed under normal growth conditions. These genes have promoters and other regulatory DNA sequences that ensure constant expression. So they're always being expressed. So first, we're going to be discussing negative operon control. So first, what is an operon? So an operon is a functional unit of DNA containing a cluster of genes under the control of a single promoter. So here you can see the operon represented in the yellow, and it's under the control of this promoter. And the operator, or the operator in this case, provides a site where repressors can bind. So the control of an operon is a type of gene regulation that enables an organism to regulate the expression of various genes depending on environmental conditions. An operon regulation can be either negative or positive by induction or repression. Remember those differences between repressible genes and inducible genes. So negative control involves the binding of a repressor to the operator to prevent transcription. So here is an example of negative control of an inducible gene. So remember, an inducible gene in this case, these are genes that are off by default but can be turned on when needed. So here, under normal conditions, a repressor protein would be bound to your operator. So this repressor protein acts almost like a stop sign and stops your RNA polymerase, your building molecule, from conducting its function of transcription. So thus no transcription occurs. But under under certain conditions, there can be an inducer molecule released, which will bind to your repressor molecule, your repressor protein, and render it unable to bind to your operator. Thus, the stop sign is now removed, and the RNA polymerase can simply go about its business. It can do its thing normally, and transcription will occur, and your polypeptide chain will form. Your protein will be coded for. Now, the inverse is true for negative control of a repressible gene. Remember, a repressible gene is a gene that is normally on. Its base state is to be on, not off. So here, under normal conditions, there's no stop sign here, and the RNA polymerase can simply attach to your mRNA, and transcription will occur, and your polypeptide chain will form. See, here's your repressor protein. But now, under other conditions, a co-repressor may be present, which will allow your repressor to bind to the operator, and thus the stop sign will now be it will be active, and RNA polymerase will not be able to bind, and transcription will not occur. Moving on to positive operon control. So operons can also be positively controlled. So with positive control, an activator protein stimulates transcription by binding to DNA, usually at a site other than the operator. So here you can see there's a different site, like that definition just said. There's an activator binding site. So it's not at the same, this binding doesn't occur at the same place as the previous example. So in positive inducible operons, activator proteins are normally unable to bind to the pertinent DNA. See, this activator protein cannot bind to this DNA, and because this is an inducible gene, no transcription is occurring. So for transcription to occur, you need this activator protein to bind to this activator binding site. This is where the inducer comes in. So when an inducer is bound to this activator protein, it undergoes a change in conformation so that it can bind to the DNA and activate transcription. So once this inducer is added to the activator protein, it can bind to the activator site, which will allow RNA polymerase to do some translation. So moving on to the positive control of a repressible gene. So this is where the opposite is true. The activator protein is normally on the activator binding site, allowing the RNA polymerase to occur. And now a molecule known as an inhibitor binds to the activator, and thus prevents the activator from binding to the DNA, and this stops the activation and transcription in these systems. As you can see, no transcription occurs. So an example of negative control it can be found when you look at lactose, the lactose operon. So the LAC operon is an inducible operon that encodes for enzymes for the metabolism of the sugar lactose. And it turns on only when the sugar lactose is present and other preferred sugars are absent. Remember, again, these are inducible genes, so these are genes that are off by default but can be turned on. And they code for genes that aid in the metabolism of lactose. And in this, for the control here is done by the LAC repressor, or LAC-L or LAC-1 binds to the operator, and in this inhibits transcription. And again, the enzymes are not produced unless lactose is present. So here you can see that process happening. So under when there's no lactose in the environment, this LAC repressor will bind to the operator, and it will inhibit transcription. So here it again acts as a stop sign, and the RNA polymerase cannot move forward and can't transcribe the molecule. Now moving on to when lactose is present in the environment, and the alactose will bind to your repressor and prevent it from binding to your DNA, which will stop transcription. In this case, because it's not bound there, transcription can happen. See, your mRNA is produced, and translation will occur following this. So in this case, it's important to know what this allactose is, and allolactose. Sorry, is allolactose is a molecule that's called an inducer. This is why the operon is said to be inducible, and the inducer is it acts on your repressor to determine whether it'll be actively bound to your DNA, or whether it'll be unable to do so. Looking at tryptophan, the tryptophan operon. So this is an example of negative control of a repressible gene. So here you can see under normal conditions, transcription will occur because these operons are normally on, but can be turned off. The molecule that turns turns it the repressor off in this case. That turns the repressor off in this case is actually tryptophan itself. So this is a nice way how the cell can regulate the amount of tryptophan by, if there is no tryptophan present, the transcription will occur, and if tryptophan becomes too abundant, it'll stop the reaction from taking place. So yeah, during this process, the negative transcriptional control of repressible genes by the TRA repressor and the operon only functions in the absence of tryptophan. So under normal conditions, when there's no tryptophan, the transcription will occur. Tryptophan becomes present, binds to this molecule, which then binds to the DNA, stopping transcription. So both of these examples, this one and the lac operon example, illustrate an important point that gene regulation allows bacteria to respond to changes in their environment by altering gene expression, and thus changing the set of genes present in the cell. Moving on to catabolite repression. So catabolite repression is an important part of global system control of various bacteria and other microorganisms. So this is regulation that is done by both repressors and activators, and results in diphasic growth when you have different carbon or sugar sources present. So here you can see the initial growth of this bacteria was really quick, and this was because glucose is used. This is a more energy efficient sugar that was used, so the organism can grow much quicker in its presence. Then this lag phase occurs, and this is when the preferred substrate, the glucose, has now become exhausted. There's no more glucose for this organism to metabolize. And then following this lag phase, there's a second period of growth, but this is slower than the first. Notice how this it sort of tapers off, and the increase is much less pronounced. And this is because the second carbon source, in this case lactose, is not as efficient for the microbe to use. So the catabolite repression allows the microorganisms to adapt quickly to a preferred carbon and energy source first, and it's achieved through the inhibition of synthesis enzymes involved in the catabolism of carbon sources other than the preferred one. So during this glucose-rich phase, or the glucose-abundant phase, the enzymes that code for the metabolism of lactose are repressed. The cell is not expressing them. And then that is so catabolite repression is the reason we see this growth curve. Just a note on catabolite repression. You might find, if you look at some other texts, some older texts specifically, that it is sometimes referred to as the glucose effect. This is actually a misnomer, as other carbon sources are also known to induce catabolite repression. So just bear that in mind. Things or caps, and it regulates in response to the presence or absence of glucose, and allows for the preferential use of glucose. Remember that I said the microbes will prefer using glucose because it's more energy efficient. Glucose can enter glycolysis without requiring as much modification as other sugars. Thus, it is more energy efficient, and cells don't like to waste energy. Therefore, when glucose is present, the genes which participate in the metabolism of metabolite repression. So how looking at catabolite activation? So for this process to occur, you need your cap proteins to bind to the DNA to aid in the efficiency of RNA polymerase and speed up transcription. Translation. And then, so here in this case, the low glucose will stimulate the release of cyclic AMP, or CAMP as it's also known, which is required for the cap proteins to bind to your DNA, because you require this CAMP cap complex. So it must, this CAMP, this cyclic adenine monophosphate, is required to form that complex. So the CA-CAMP complex will increase the affinity for RNA polymerase to bind to DNA, thereby increasing transcription by approximately 50 times. I read in a certain source. And an important feedback loop that's present here is high glucose in the cell will lower your CAMP. Consequently, low glucose will lead to an abundance of CAMP. So here, low glucose will increase your CAMP. Thus, in bacteria, the level of CAMP or CAMP varies depending on the medium used for growth. In particular, CAMP will be low when glucose is the carbon source. So how are these CAMP molecules regulated? So CAP activity is modulated by the CAMP, as we just said, but levels of CAMP controlled by adenyl cyclase, which converts ATP to CAMP and PPI by PEP. This PEP is phosphoenol pyruvate, and will be active only when little or no glucose is present. When you continue on in biology, you'll learn more about this molecule. It's an energy-rich molecule that's used quite a lot. And then in the absence of glucose, the CAP proteins is active and promotes the transcription of operons used for the catabolism of other sugars. Again, allowing the bacteria or the organism to use sugars not when glucose, its preferred sugar, is not present. So how is this done? You can look at this cycle down here. This is how the CAMP is essentially produced, because it requires this phosphor. So the PEP, this phosphoenolpyruvate, this is called PEP group translocation, also known as the phosphotransferase system. This is the method that is used by bacteria for sugar uptake when the source of energy is from phosphoenolpyruvate. So this PEP will donate its phosphor across this cycle, which will then end up being used by this enzyme 2A to produce your CAMP molecule. And then when glucose is available, this enzyme will instead donate this phosphor to the glucose to phosphorylize the glucose. So thus this will not be produced. But when glucose is not present, the phosphor is free to be transferred and the CAMP can be made. So looking at what this means for the microbe in the instance when lactose is present but there's no glucose, looking specifically at the lac operon and lac repressor and CAP. So here you can see the CAP is bound to the CAP site, and the CAMP molecule is present, and it's present because there is no glucose, so that phosphor, remember, from that cycle is now available and CAMP can be produced. So this is bound. The presence of lactose also means allolactose is present, so the repressor is inactive. It can't bind to the operator, so you see the operator does not have the repressor on it, so thus transcription will occur. So this is the condition when you have lactose and no glucose. So what happens when we have both lactose and glucose? So in this case, the cell will favor glucose. So the genes required for the metabolism of lactose, we don't want them to be synthesized, to be made here, because it would essentially just be a waste of energy. So the but glucose, lactose is still present. So this allolactose will also still be present. So your repressor will be inactive. But remember now that CAMP molecule cannot be produced because the phosphor group is not available. So CAP will not be on the CAP side. So transcription will be inhibited by the lack of CAP, those proteins. What happens when neither lactose nor glucose are present? So because there is no glucose, your CAMP complex will be on the CAP site. This CAMP will be made because there's no glucose to accept that phosphor group, but there's no lactose, meaning the allactose that we had present over in these two examples will not be present. So your repressor will be able to bind to your operator, so transcription will be blocked by the repressor. And in our final example, what happens when there's glucose but no lactose? Again, here, obviously, we're going to want the glucose to be metabolized. So CAP will now be inactive because the phosphor group is used to phosphorylize the glucose. And then transcription will be inhibited by the lack of CAP and the presence of our repressor, because allactose, remember, no lactose present, so allolactose is not present, meaning the operator can be blocked by the repressor and the CAP will not be active. So in this case, it's blocked on both sides. In that, you can view it in that way. So transcription will be inhibited by the lack of CAP and the presence of a repressor. So moving on to just a few review questions again on today's lecture, similar to the ones we had last time. So which of the following is not a binding site on a ribosome? The A site, the B site, the P site, and the E site, or none of the above? I'll give you about 10-15 seconds for this. Okay, hope everyone is done. The answer here, so remember the question asked which one is not a binding site, so the correct answer would be B, the B site. Just as a reminder, your A site is your aminoacyl or acceptor site, and it binds your incoming aminoacyl tRNA. Your P site is your peptidyl or your donor site, which binds initiator tRNA. And your E site is your exit site, which briefly binds your tRNA before it leaves the ribosome. So my second question, what is the function of the Shine-Dalgarno sequence, that AGG during protein synthesis? Does it bind the 30S and 50S ribosome? Does it terminate protein synthesis once completed? Does it provide the tRNA with binding specificity during elongation? Or does it interact with the rRNA molecules and anchor the 30S ribosomal subunit at the correct location on the mRNA template? Again, just 15 seconds or so to answer, to respond. Okay, and here the correct answer is that it interacts with the RNA molecule and anchors the 30S ribosomal subunit at the correct location. Remember, it played a role in forming that initiation complex where it essentially gave the 30S ribosome instructions as to where it should be starting on the mRNA. So the third question, what is the definition of an operon? Is it a cluster of functionally related genes that are controlled by a shared operator, a regulatory protein that binds to pieces of DNA, an enzyme which plays a role during the formation of the CAE, the CAP-CAMP complex, the enzyme responsible for the regulation of sugar uptake in prokaryotes, or none of the above? Again, just 10-15 seconds. The correct answer here is A. It's a cluster of functionally related genes that are controlled by a shared operator. Now, our final question, I think this is a bit of a gimme. So true or false, in bacteria, the level of CAMP varies depending on the medium used for growth. I'm going to give you guys about 10 seconds for this one because I think it's pretty easy. And the answer in this case is, of course, true. So that concludes our lecture for today. Thank you guys for having me for these 2 guest lectures, and I hope you learned something.
Okay. Any questions for somebody? So I would say Dr. Peterson did a great job for explaining us for the 2 lectures about bacterial genetics. And so just please, I think it's recording right now, and just please send me the slides.
I will
And
definitely.
yeah, and I hope you have a good semester. And I think next semester you might be teach this class. So it's I think you're a very good teacher for these parts, and I think all the best wishes for you.
Thanks, Doc. I appreciate it. Thank you guys for having me.
Thank you.
Thank you.